this approach was suggested to regret not by us, but we use it. Important is that we don't have absolute values, we have just relative turnover rate. It means I cannot tell that the turnover rate of this soil, for example, is 30 years or 100 years, but I can conclude that this soil has faster turnover rate than this soil because this inclination is strong. So this approach was suggested initially just for carbon, but we thought if this worked for carbon, maybe it worked also for nitrogen. So we can also present logarithm of natural isotopic composition of nitrogen to its content, and maybe we'll get also similar results. So I will present such results from such studies, but at first maybe a few words how isotopic fractionation, changes of isotopes is ongoing. There are substances which have light isotopes, for example, carbon-12 and carbon-12. But there are some substances which have, naturally about 1%, have heavier isotopes. And you can imagine if the energy of the substances is the same, then these substances will be moved slowly because of the kinetic energy. And there are many options how they can move on this way. But important is that because the red points which reflect here, the heavier substances move a little bit slowly, then there are some processes ongoing that, for example, if there are some membranes, for example, by photosynthesis, for example, the cell wall, then lighter substances with lighter isotopes will move through this membrane a little bit faster. They will also go into the chemical reactions a little bit faster. And this is presented also that smaller will move faster, for example, through some open doors. This is just an example. And the results of this, they are also very well known and used, for example, if water will be evaporated, then in vapor, there are some less heavier deuterium. So that in water, it is more heavier compounds. It is similar, for example, in water and O18. Here it will be less O18 in the vapor. The same for bicarbonate for the dissolution between the gas and the water. And also, for example, if calcium carbonate will be dissolved, it will be more in solution because it's going easier into the solution compared to the solid. This is just as example. We collected such data and just last year made such a meta-analysis how we can use the isotopic composition of carbon and nitrogen to assess soil organic matter turnover. So we looked how litter will be transformed to soil organic matter and how the changes of isotopic composition is ongoing in the depths. But the first, maybe what is also important, we tried to assess mechanism of carbon-13 and nitrogen-15 enrichment and depletion in soil organic matter and leading to the changes of isotopic composition, which will be frequently termed as discrimination. It means if this is, for example, 0 line for the sources, and then from the source, from organic material, microbial biomass respire something, then carbon in the CO2 will be depleted. So it means in this direction, it is going depletion, processes leading to the depletion of carbon-13. In this direction, it's going to the enrichment of isotope. So for example, if microorganisms use organic substances, then the CO2 will be depleted but the residue, the formed microbial biomass of soil organic matter, will be then enriched. The same for nitrogen, but today we will focus only on this part of the processes. So we collected data from literature. These are not our data, from various parts of the world for forest, grassland, and shrubland. and actually they are located in all biomes except tundra. And then it was about 150 soil profiles with isotopic composition and soil organic carbon and isotopic composition of nitrogen and total nitrogen. It means we had such soil profile in which with the depths it was presented isotopic composition of carbon and isotopic composition of nitrogen. And these are exactly the same pictures which I presented you before. From these lines, we calculated this inclination. And additionally, we also collected many soil profiles from which, from the litter, soil organic matter will be formed. And maybe we can conclude also about something about the initial step of the soil organic matter formation from litter. Let us come to the results now. In the first slide, I would like just to present how from litter soil organic matter will be formed. You see here the isotopic composition of carbon, difference between soil organic matter in mineral soil, and isotopic composition of litter. Here, the same for nitrogen. This is one-to-one line. And you see that all points are located above the one-to-one lines. On the average, it's somewhere here. It means that actually discrimination for carbon is much less compared to the discrimination by nitrogen. It means nitrogen will be lost. Maybe it's leaching, maybe these are some gases, but it is important conclusion that discrimination of nitrogen from the same compounds, from the same plant residues is much stronger compared to the discrimination of carbon. And it means that the nitrogen turnover is ongoing faster than the turnover of carbon. Here it's presented the same, but between the difference between the topsoil and the subsoil. This is for carbon, this is for nitrogen. And again, you see that the most points are located above the one-to-one line. And this is also confirmed that also in the soil, nitrogen turnover, if you compare this value to this value, is about 2 times faster than turnover of carbon. It means we have interesting situation that from the same compounds, from the same soil organic matter, nitrogen will be turned over faster than the carbon. Let us look a little bit further. Now we can look for the inclination, and here it's presented inclination for carbon, and here's for nitrogen. It means each of these points is actually just the inclination of such a regression line from many points. And you can see again that the most points are located above the one-to-one line. And the discrimination for nitrogen is about 2 per mile. For carbon is also 0.9. It means it's about, again, 2 times faster. How it is possible that nitrogen will be turned over in soil faster than the carbon? I think there are many reasons that's possible. At first, carbon, when it will be mineralized, it will be lost in the CO2 and will leave soil. But nitrogen, when it will be mineralized, it remains in soil and will be reused again and again. It means nitrogen reutilization by microorganisms. This is one reason. But second reason is probably that the nitrogen compounds will be decomposed faster. For example, proteins, amino acids, DNA, RNA, they will be decomposed faster compared to the organic compounds which do not have nitrogen. Maybe there are some other mechanisms, and maybe some of you can also suggest some other mechanism why nitrogen will be utilized in soil much faster than the carbon. It means if we're trying to present this as a picture, actually we have the following situation that for nitrogen turnover, there is turnover between the plants taking nitrogen, microorganisms mineralizing nitrogen, and then nitrogen is going back to the plant. So this cycle is comparatively short. However, if we look for the turnover of carbon, after each mineralization, part of the carbon, not part, all of the carbon is going into the atmosphere, will be mixed with the initial isotopic composition of CO2 in the atmosphere, because in the atmosphere is huge pool having this isotopic composition, and then coming back by the photosynthesis of plants, it means the cycle of carbon is much larger and much bigger. This is one of the possible explanations. Let us look now for effect of some factors. One of the factors which is very well known is the temperature. You see here again the inclination of the lines which I presented to you before for carbon and for nitrogen. And we can conclude that both lines are ongoing nearly parallel together, but nitrogen is much higher. Regression line for nitrogen is much higher than the regression line of carbon. It means nitrogen turnover is faster, but the increase with temperature is nearly the same. If we look, however, for mean annual precipitation, then the inclination is much less, and for nitrogen it's actually not significant. It means that the effect of temperature for the turnover of carbon in soil is much more important than the effect of the precipitation. Let us look now for the biomes. You see here climatic conditions for temperate, Mediterranean, subtropical, and tropical soils, and this corresponding temperature. And actually, I hope it is very clearly to see that with the temperature, actually the turnover of carbon is increasing. Similarly, also for nitrogen. For tropical soil, we had just a couple of points, so there are no conclusions here. But it means again that in the subtropics and tropics, the turnover of carbon is about 2 times faster than in Mediterranean and temperate climates. And again, if we look on the increase of temperature, probably the temperature is the most important factor. If we look for the effect of forest types, evergreen, deciduous, coniferous, mixed forest, then we can recognize that in the coniferous and partly in the mixed forest, the turnover of carbon is slower compared to the evergreen and deciduous forest. And probably there are 2 mechanisms. One mechanism which I mentioned before is the temperature, because surely the temperature in evergreen and deciduous is usually higher than in coniferous and mixed forest. But the second is the litter quality. Coniferous litter will have a lot of lipids, a lot of lignin, and they will be decomposed more slowly, and consequently also turnover in soil is also slower. For nitrogen, there are no clear effects, but there are only few data for nitrogen in these ecosystems. Let us look now for another factor, for the CN ratio of the soil organic matter, because many studies showed, and usually we also think that closer is the CN ratio of soil organic matter, the faster is also the turnover. For carbon, it is very clear. It is faster turnover with the, it is slower turnover for increasing soil organic matter. For nitrogen too, but the regression was not significant. And again, I think the mechanism for that is the quality of litter, which direct affect the turnover. And this is temperature, which is indirectly affect the turnover. Let me now to summarize all the pool of the mechanisms how carbon and nitrogen turnover in soil can be assessed and which mechanisms contribute to the changes of isotopic composition. If we have here the depths and here isotopic composition of carbon and nitrogen, so we have usually such curve for carbon and much stronger discrimination for nitrogen 15. The first step by the formation of soil organic matter, this is carbon which is coming from the roots, and roots are usually heavier isotopically than the shoots, and consequently litter coming from the shoots has more negative value compared to the litter which will be formed in the Dept soil coming from the soil organic matter. This is presented partly here, so the roots are heavier than the shoots. And also, if we're speaking about nitrogen, so all data for nitrogen will be presented in green. For nitrogen nodules, for example, of the legumes, they are heavier than the shoots. It means this is also the nodules nitrogen fixation by rhizobia also contribute to the stronger discrimination of nitrogen 15. Another mechanisms are that by the assimilation of plants, by the uptake of nitrogen from the plants, plants take mainly ammonia and nitrate, and they are already slightly depleted. If they are depleted and will be taken by the plants, then heavier compounds remain in soil, which also contribute to this line. And also the same by the mycorrhiza fungi. Mycorrhiza fungi can take also lighter isotopes or lighter compounds with lighter isotopes, and consequently more heavier remain in soil. Carbon soil organic matter will be utilized by microorganisms, and CO2 will be released, and also for nitrogen by denitrification nitrogen 2 and N2O will be produced, which are slightly depleted. If they are slightly depleted, then heavier remains in the soil. There are many mechanisms how microorganisms contribute to this. So if microbial transformation is ongoing, then heavier compounds remain in soil, lighter go into the atmosphere. Another contribution is about by Zeus effect. We can speak about this in discussion. The processes of humidification and stabilization of organic matter on clay minerals also contribute to the heavier isotopic composition, because certain C-depleted compounds will be preferentially absorbed. Nitrification, ammonification, and sorption of nitrogen-15 and enriched ammonia also contribute to the discrimination of this line. Depleted nitrate will be leached, and consequently heavier again remains in soil. The turnover of carbon and nitrogen will be more slowly with the depths, but nevertheless it is ongoing, and therefore the inclination of the line here is much stronger compared to the inclination of both lines in the depths. And in the depths also it is important that DOC, dissolved organic carbon and dissolved organic nitrogen, will be also migrating, and this is heavier than the soil organic matter here, and consequently by the absorption this contributes also to the increase of the isotopic composition of carbon and nitrogen with the depths. So I would like to come to the conclusions. I think this is very interesting and, in my view, very simple approach to use just isotopic composition of carbon and logarithm of soil organic carbon content. And this is just of one of many methods to assess the soil organic matter. To regret, and this is the biggest problem, that this approach delivers only the relative turnover rates. For the first time, we made such meta-analysis of the application of this approach for various soils worldwide. And the new here that we, for the first time, use it to assess the nitrogen, to assess turnover of nitrogen in soil based on this method. And it is interesting that the turnover of nitrogen is about 2 times faster than the turnover of carbon. And I think this is related with the situation that nitrogen will be recycled in soil, but carbon by each cycle will be at least about 60% or even 70% will be released back into the atmosphere. So the turnover rate in the boreal forest are slower than in Mediterranean and in subtropics, and coniferous and deciduous are similar, but evergreen forests have faster turnover rates. And actually, the temperature is probably the most important factor, not the precipitation. Turnover is getting slower with the increase of carbon and nitrogen turnover rates. But I think it is also very important that in the whole paper, we have tried to summarize all the bunch of the known mechanisms of carbon and nitrogen fractionation in soil between plants, microorganisms, and soil. And before I thank you for your attention, you can take a look again for this paper because all the things are much better explained there compared to my talk. Thank you very much for your attention, and I'm open for your questions.
Thank you. Thank you very much for this interesting talk. It opens a lot of possibilities of studying soils and the turnover of organic matter. But not only this, I think that for paleopathologists, this issue is also very important. So please, your questions in Spanish or in English, what you want, we can help. Ya tiene. I need to translate.
Okay.
He can easily.
He can, but he is very shy.
Young girl is very shy.
Yeah.
Muchas gracias por la plática. Realmente es muy emocionante, ¿no? Ver cómo se construyen estas figuras en tiempo real. Entonces estoy realmente emocionado por eso.
He is very exciting and grateful because this kind of presentations give the models to be more robust, something like that.
Ok. Una de las premisas de las que se entiende por lo cual el carbono más profundo es más antiguo o se puede considerar más antiguo o más pesado, muchas veces se tiene, o como intuitivamente se tiene la idea de que es porque es el residuo de la descomposición in situ. Pero esto que nos muestra de la migración del carbono orgánico disuelto que va incrementando su peso isotópico es una aproximación diferente porque nos permitiría entonces discriminar el carbono antiguo del carbono reciente, pero de origen más pesado por efecto de especies. ¿Sí me entendí?
Voy a tratar de traducirlo como lo dijiste y si no me paras.
Okay.
So this situation that the heavier isotope goes down because of the age of the organic matter. This is one of the possibility. But the other issue is that the dissolved organic carbon can go with this heavier isotope. Then this opens a new possibility to evaluate how the carbon is moving in the soil. That's right?
Right.
Yes. So
La pregunta era, bueno, no la terminé porque me quedé pensando. Esto puede no ser lineal si especies diferentes generan diferente discriminación isotópica en la producción de esta materia orgánica disuelta, posiblemente por exudados de raíces. ¿Sí?
So this opens the question that different species produces different type of carbon and the discrimination of each species is different. So how to evaluate this?
So thank you for your question. And actually, there are two questions. The first question is related with the migration of dissolved organic matter, dissolved organic carbon and nitrogen. The second is related with the assimilation of carbon by various plants. So the first one is, yes, dissolved organic matter migrate, but this dissolved organic matter usually is heavier than the local soil organic matter from which it will be removed. It means if we find dissolved organic matter here, probably it's migrating from somewhere from the top. And there are two reasons, at least in my view, there are two reasons. One of the reasons is that lighter dissolved organic matter will be easier decomposed. The second reason that the roots which produce a lot of rhizodeposits, which are partly soluble, and also when the litter will be decomposed, they are also partly heavier dissolved organic matter. And consequently, this is dissolved organic matter is heavier and going into the migration, and this contribute also to the inclination of the lines of these two lines in the depths. The first question, what you mentioned, is about the assimilation by the plants and the formation of various compounds. Please have in mind, this approach, maybe I have not mentioned this at the beginning, is based on the steady state. It means over the many years, actually decades or even longer, there is some kind of the steady state, so steady level, but changes in the processes. So this is like a river. When you go to a river, the river has the same level, but it's continually flowing. So this is a steady state which we have also very frequently with soil organic matter, carbon or nitrogen and soil, that the level remains very long the same, but the processes are ongoing. And what we measure here, it is situation over a very long period of time. So it means this approach works if there are no changes over the longer. So if you have vegetation and soil like in steady state, only if you will have land use changes from forest or grassland to cropland, then the composition of the organic compounds which are coming on the soil and rhizodeposits coming into the soil, they will be probably very different compared to the initial time when the soil organic matter will be formed, and then we will not get such lines. So it makes sense to use this approach only for the soils which are long-term in the steady state with the vegetation. Thank you for your questions.
Thanks for much.
Otra pregunta.
Hi, thanks a lot for your talk. It was very nice and interesting, and I'll have a look at the paper. My question is regarding wetlands, because they have very different, well, with the water in an anoxic environment, and also between freshwater and saltwater wetlands, it will probably be different, no? Because freshwater wetlands produce a lot of methane, while saltwater wetlands don't. So I'm hoping maybe you will be interested in doing something like this as a meta-analysis.
Also very interesting question. So in wetlands, at first in wetlands, so all the data which I presented, it's only for uplands. It was already when we selected the papers for this meta-analysis, we selected only the papers from the uplands, that they are not overflowed. For wetlands, there are at least two interesting issues. One issue that because processes in wetlands are ongoing much slower than in the presence of the oxygen, the isotopic discrimination should be stronger. But there is another problem, that in wetlands, lignin will be not decomposed, or will be decomposed much, much slower compared to cellulose, proteins, and other compounds. And because it means it is more processes there, which I cannot just evaluate here now in the short talk, but we can think about this. So it means from one point of view, the discrimination will be stronger. From the other point of view, some compounds will be decomposed and disappear. The other compounds like lignin will remain. And lignin in the plants is strongly depleted. So lignin in shoots and in the roots and in the leaves is usually strongly depleted compared to the average isotopic composition of the full plants. And I cannot tell you now in just a minute what could be the result of these both opposite processes.
Thank you.
Osis.
Yeah, thank you, Professor. It's really interesting topic. My question is a little bit out of this presentation. It's more related with another environment, like arid and semi-arid environment with the formation of pedogenic carbonates. I don't know if you think it's interesting to evaluate the turnover related with this precipitation of pedogenic carbonate and make a comparison with the organic matter in this environment, different ones. Thanks.
It would be another talk
Yeah.
to
Sorry.
the pedogenic carbonates, and this is another interesting issue, that the isotopic composition of lithogenic carbonates is about 0. Yes, isotopic composition of pedogenic carbonates, because of this CO2 in the soil, is about -14 on average, depending on the vegetation, because in arid climate there is a lot of C4 vegetation, which can also lead to the final results of the calcium carbonate of about 0. So it means it is, if you have lithogenic carbonates and pedogenic carbonates as a mixture, and only from C3 vegetation, not C3, C4, and you know when the process is started, then you can estimate the turnover rate of pedogenic carbonates. But usually you don't really know when the process is started. So you have changes, but you don't know when you don't have the timeline. If you have special situation, for example, one of the colleagues of Sergei, Konstantin Pustavoitov, work with such changes in which he knows the time when the changes occur. Then it's possible to estimate.
Thank you.
All right, thank you very much. And yeah, it's very nice to finally meet you. I mean, I have read some of your papers, but it's nice to put a face on them. So my question is actually... My face corresponds. Yeah. So my question is very similar to what the previous person asked, because I've been working with calcareous soils. So I wanted to know, you mentioned the relation of temperature and of humidity, right? But what about pH? Like if you have like more acidic or more like basic environments, how does this affect the turn?
So we checked in this database the relation to pH. We did not find any significant changes of the inclination, neither for carbon nor for nitrogen. So therefore, I have not presented this.
Sergei, ¿quieres preguntar algo?
Sí.
Es que vi que levantaste tu manita.
Yes, but of course I had to give chance for real specialists in isotopic issues before poor paleopedologist could ask his questions. But really, this talk has also very important paleopedological and paleoecological issues. And if I'm allowed 2 questions. Great. Even 3. Great. Maybe even 3. One is quite particular one. Could we go to the slide, one of the first ones, where you showed us the cases of changes of δ13 with depth?
Just a second.
It was in the beginning.
Yes.
This typical curve of increase of δ13 values. This one. This is very interesting that exactly such graphics we find in many cases, numerous papers, concerning the soils of Yucatan Peninsula and other areas of Maya region, of ancient Maya culture. By the way, there on your map there is a big white spot there, so we could feel it. If you like.
Sorry for that. Yes, let us do together.
But even in the literature, there are very similar curves like this, especially of our North American colleagues, and they interpreted in a very interesting way. For them, this is direct evidence of change of plant cover from the earlier, much heavier, ancient, much heavier values, which are preserved in depth, to the recent, much lighter values, which correspond to present-day rainforest, tropical rainforest vegetation. And why it was heavier earlier? Because of maize cultivation during the Maya time. The difference of values is more or less the same from about minus 2627 to minus 20, for example. But interpretation, of course, is completely different. For them, it is a paleoecological indicator, an indicator of plant change, plant cover change, and not related to the decomposition, recycling, and these issues. What do you think about this situation? Are they not are their conclusions not correct? And are there some instruments, isotopic instruments, which you could propose to check whether this is just because of decomposition or really plant cover change took place?
So I fully agree that if there is changes of plant type of plant photosynthesis, C3 or C4. So you mentioned maize was cultivated there. It was
typical C4.
It's a very typical C4. It has usually isotopic composition -12, -15. So in between, depending on the water content in soil. And surely, if you have changes of the vegetation photosynthesis type, you cannot use this method. Then you use it just to interpret changes of vegetation. So it means this is always what I presented if there are no vegetation changes. In your example with the maize cultivation in the past, the big question is how intensive was this cultivation? Was this continuous maize? Was the productivity similar like now? I guess it may be 10 times less, maybe 20 times less than today. And it means that also it was not really C4 soil, probably. It was a mixture between previous forest and then maize, and then new crops today, which are probably C3 like wheat or similar. So the interpretation will be very difficult because you don't know how long maize cultivation was, which portion of the soil organic matter from C4 signature was exchanged with from C3 signature was exchanged by C4 signature. But I know such situations are possible. They surely give information that it was other vegetation. And then it's a question how to interpret this other vegetation. The question related what are other approaches to be sure that this is maize cultivation. This is actually the residues, or these are not necessarily isotopic approaches. These are residues of some seeds, some plant residues, which are maybe some pollen, maybe some phytoliths. I'm not specialized on this. I cannot answer. I know that the paleo people use very broad range of the biomarkers and also of various approach to reconstruct the previous paleo environment. And I know that they have very broad fantasy.
This is right, but in these cases, in these published cases, usually these paleo soil people do not have enough results for their fantasy.
No.
The soils of Yucatan, and these are mostly surface soils, do not contain much pollen. Of course, all macro residuals are decomposed, and even phytoliths are very few.
Because limestone is
I cannot give now any suggestions to develop your fantasy. I'm sorry for that.
I hoped so much that you will give us some instrument to discriminate between this fractionation and making the signature heavier due to due to vegetation change versus decomposition and
Sergey, I suggest we take a seat together with a piece of paper and think what is possible. I cannot really answer your questions now, how it is. We need to look for
I
some
think
data.
we could discuss maybe seeing also these papers.
Thanks.
Still a chance for the second question, or you are not allowed?
One moment. ¿Hay alguien que quiera hacer una pregunta o le dejamos a Sergey que siga con el micrófono? Creo que puede seguir con el micrófono.
Very well. This is a general question of a person quite ignorant in issues of poor paleo-pedologist, ignorant in issues of dynamics of soil organic matter. I am really impressed by this result that nitrogen cycling is much faster than carbon cycling. And I have a certain discrepancy in my mind concerning the information which we all got when we studied soil organic matter, being soil science students, from the textbooks. There we learned that soil humus, the most advanced and decomposed components of soil organic matter, are enriched in nitrogen.
Yes.
Then I would think that nitrogen components, components rich in nitrogen, are more stable. And now I hear from you the idea that it is, on the contrary, nitrogen components are recycled much quicker. For me, it is a sort of discrepancy, but I hope you have an explanation.
It's an interesting question, very interesting. We also thought about this. So I think there are some reasons for that. One of the reasons is that I fully agree that the CN ratio in the soil organic matter is about 2 times, 3 times smaller compared to the initial plant residues. One of the reasons for that are amino sugars. Amino sugars are the parts of the microbial cells which will be incorporated stronger because of amino groups to the negatively charged clay minerals. This is surely, and also organic matter groups. Yes, this is, but please have in mind the stability and turnover is not always the same. It means we can have stable part, which is very stable and consequently has very long turnover time. And we can have also faster part, like amino acids, proteins, which will be utilized by micro DNA, which is also a lot of nitrogen there, which will be utilized much faster. So I think it is like decoupling. Some compounds of some nitrogen compounds will be stabilized, mainly amino sugars. Other compounds, probably like residues from proteins, amino acids, DNA, they will be utilized, maybe mineralized more faster, and then will be included in the next cycle.
Thank you very much. Very interesting.
So just related to the same question that Sergey said, I'm also not an expert in organic matter, but seeing this, that you concluded that the nitrogen turnover is more than twice as fast than the carbon turnover. Does this mean that denitrification processes are happening at a lower rate to lose nitrogen to the atmosphere than oxidation of organic matter to produce CO2? Is that the way to read it? Can you comment a little bit about that?
Just a second. Yes. So, I think it is surely that when carbon initial organic compounds will be mineralized, then about 60% and over more than 1 year, it's 90% or more going to the CO2. From the denitrification, this is just few percent. And we usually think about the denitrification in agriculture soils, in which denitrification is maybe 10%, but actually maybe about 5%. It means we cannot compare this 60, 70, or even 90% with this just 5%. So the losses of gaseous losses of nitrogen per unit of nitrogen is order of magnitude less compared to the CO2.
Okay, thank you very much. I have to finish here.
For the great questions, this also pushed me a little bit to think.
Yeah, it was exciting. And thank you very much for coming here. And I give you this certificate. Y muchas gracias a todos los demás por haber participado. Y pues seguimos con el programa del simposio.